Hello everyone. Today we are going to discuss on the topic of the DNA replication. DNA replication is a biological process of producing two identical replicates of a DNA form from one original DNA molecule. DNA replication occurs in all living organisms, acting as the most essential part for the biological inheritance. Unwinding of DNA and synthesis of new strands take place by an enzyme known as helicase, which is result in a formation. DNA replication occurs during the S stage of the interphase. Then we discuss of the replication process. The DNA replication, including all the biological polymerization process, in which three enzymatically catalyzed and coordinated step. One is initiation, second is elongation, and third is termination. Then we started from the initiation. Initiation is a cell divided. It is the first step of the DNA replication. Division of initiation in a pre-replication complex sequences. Binding of a DNA protein to on a E. coli bacteria. Further, the DNA helicase is loaded as a paired of a template strand. Primer bind to the chromosomes. Synthesis of RNA primer. Initiation of the DNA polymerase by DNA polymerase. The next is elongation. DNA polymerase catalysis is a step-by-step addition of the nucleotide unit to the DNA chain, and it has a 3' to 5' activity. Each growing fork strand called as a leading strand, and it is synthesized continuously from a single primer on the leading strand template. DNA polymerase then adds the piece of the DNA called as the Ozaki fragment to strand between the primer. Involves the addition of a new nucleotide based on the complementary template strand. Then the next is termination. Once both the continuous discontinuous strands are formed, our enzyme called as a exonuclease remove all the RNA primers from the original strands. Once completed the parent strand, and it is complementary DNA strand coil into the family double helix shape. Then the next one is enzyme involved in a DNA replication. The first one is DNA helicase. It is used in unwind and separate the double stranded as a move along. The next is a DNA primerase, a type of RNA polymerase that generate DNA primers. Then the DNA polymerase, synthesis of a new DNA molecule by adding the nucleotide by lagging the DNA. DNA gyrase, unwind and rewind DNA strand, and DNA ligase joins DNA fragment together by forming the phosphodiester bonds between them. This is all about the DNA replication. Thank you.